So thanks for giving a wonderful introduction to G by E and stability, Lucia. So I'm going to talk a little bit more in this presentation about how can we actually use stability metrics in breeding programs, because there's lots of work that's done in characterizing G by E and stability within breeding programs, and we know that breeding programs are interested in developing varieties and releasing varieties that have, for instance, high yield, but also stability in that yield across environments. And it's kind of a little bit of an under-researched area. And so what I'm going to be presenting is a little bit of work that we've been doing in this space. So just to start off with, breeding populations that a lot of you work with they contain lots of variation in stability. And really that's a function of the fact that we have genotype by environment interactions occurring. So the reaction of genotypes to different environments are unequal between genotypes. They change in how they react to different environments, and they change in their ranking. And so there's two ways we can really express that. So in the plot on the left here, we're showing a population that varies in their mean performance across environments, and then also in their stability. And here we're expressing stability as kind of an absolute value or a variance. So the higher the value, the less stable the genotype is. We can also express it like as in an equivalent way, but as reactions to environmental covariates that are either known or latent in the data. And so a really big reason why that's useful to express it that way is I've just highlighted two individuals in this population, and looking at the plot on the left, you would think they're quite similar in that they have a similar mean performance across environments and a similar value for stability. But if you partition out the way they react to those environments, so instead of taking an absolute value but look at their responses, you can see they'd have different responses for a regression on that covariate. And so that's just something that to keep in mind as I talk through this presentation. So what I'm looking at is how can we actually leverage this genetic variation in stability within breeding programs. So most of the research up until now has examined this question kind of in terms of a passive selection of stability. So we have a population that we've collected phenotypes on, and we've done an analysis, and we can see there's G by E in this population, and we can estimate some stability metrics. And really the question that we're asking is, should we release line A versus line B? But there's been very little research in terms of how can we actually generate stable products. So that's more thinking about which genotype should I cross line A with line B in order to create high-performing and stable products. And really, if you think about what breeding programs do, we're in the business of actively making crosses and creating improvements. So this is something that deserves some attention. So what's the current prevailing sort of view in terms of how we treat stability metrics in breeding programs? So most suggest the use of kind of an index approach where we have the average performance of a genotype across environments, and then we apply some penalty that's basically how unstable that genotype is. So if we're trying to make a list of genotypes and we want to basically promote individuals that combine high performance and stability. And so we can express it in an index like this. And so this makes sense if we're thinking about releasing varieties for release, because it is promoting individuals with high mean and high stability. But the thing I'd like to highlight and look through in this presentation is whether that actually makes sense for crossing in a breeding program, whether that's for a population improvement component or for developing products. So I'm just going to work through a really simple example here that tries to highlight why this might be a problem. So I've got 100 genotypes in this little toy example. And so they've got variation in their mean genetic performance across environments, and then variation in their sensitivity to their genetic values. And I'm expressing that as a slope or a regression slope on environmental covariate, exactly like in the previous presentation. And so what we're going to try and do is pick two pairs from this population of 100 to create progeny or products that combine high performance with high stability. And I'm going to look at three different ways we can select them. One is just selecting on the mean performance across environments. One is using that index I showed on the previous slide, so a penalized mean. And then the other one we're going to call decross, and I'll explain that later. So this is the population we have. And so on the y-axis, we're looking to push up that mean genetic value. And on the x-axis, I'm showing the slopes of regression. So the bigger the number, the more unstable or the more sensitive that genetic value is across environments. So we're really aiming for a target up here. Push up the mean as far as possible and aim for a stable genotype relative to the population we have under study. So if I was just to pick individuals based on the mean only, I would pick these 4 genotypes and cross them. And so you can see we maximize the response in the mean, but we're ending up with progeny that are less stable relative to these parents. So they'll on average have, I'll just note the average here, and you can see it's a value of about -2. So if I was to use a penalized index, I'd pick these 4 individuals. So you can see we're no longer picking those extreme genotypes that have large values for their slope, because we're wanting to choose individuals that combine high mean performance and stability. And if we look at their progeny, you can see that we're going to take a little bit of a hit on the mean, but we'll end up with more stable individuals. And the question I'd like to ask you is, can we do a better way of doing this? Is there a better way of utilizing the genetic variation in this population so that we can push up the mean genetic value and also create stable genotypes? And so the approach that we're suggesting here is to be more strategic about how we make these crosses. And so this is what all this approach is doing is recognizing, and we're dealing with additive genetic values in this case, is that we can, instead of penalizing individuals that are unstable, unstable, we can utilize them by strategically crossing them with genotypes that have complementary patterns in their G/E patterns. And so these 2 individuals that both have a high mean, but they're individually unstable, we can cross them together, and if the expectation of their progeny average is essentially stable, so their progeny will be stable. And so you can see here, if we're a bit more clever about how we design these crosses, we can get more response in the mean and more stable individuals. And so we're getting about the same as what we'd get if we selected on the mean only, and we're even doing better than if we selected based on that index. So how does this work? It's really simple. It's quite similar just to using an index, but instead of focusing on the parents themselves, we're focusing on the outcome of the cross, so the progeny of the parents. And so the reason that's useful is we don't discount individuals who are unstable, because really their instability is really reflecting just adaptive ability to certain environments. And so what that means is we're able to leverage the total genetic variation that's available in the population. And so all you do is calculate the mean and stability for all of the possible crosses that you have in your population and pick the best ones. And that's all I was doing there. It's completely analogous to if you're thinking about regression on environmental covariates, whether they're latent covariates or known covariates, simply matching genotypes that just have complementary slopes for those G/E patterns. So I'd like to test how this works using simulation. And the question I'm going to ask is, if my goal is to create stable products that I would like to release as a breeding program, does this crossing strategy outperform crosses based on the mean only or based on a penalized mean index? And then I'm also going to look at how does this depend on the number of environments and the level of G/E that I have in the program. And I'm using an inbred breeding program for this example. So how does the simulation look? So it's quite simple. I just start off with a breeding population, and from that population, exactly like the example that I showed you before, I'm going to use these 3 different approaches to create products and then evaluate the performance of these 3 different strategies. I'll then undergo a cycle of population improvement and basically repeat the process and do it 5 times. Just to keep it simple, I'm not cycling any of the products that I make back into the population improvement component, and that's just to make sure that these comparisons are fair. So really what you can think of this is we're all we're trying to do is take a population and cross it in such a way to create products that we release. So we're not necessarily thinking in terms of genetic gain. So how do I evaluate the performance of these strategies? Well, G by E is all relative to a population, and the interactions that you get is all relative, so you have to compare it to something. And so what I'm doing is I'm estimating genetic values for stability in the breeding population from which we're going to create the products, and then calculating, basically getting a distribution of genetic values for stability within that parent population. And so then you can slice it up into different percentiles and say, I'm going to try and create a product that's in the top 1% for stability relative to the breeding population or the parental population. And so we can compare for different criteria that we're aiming for stability, which strategy will maximize the mean performance given my requirement for stability. And so in terms of results, this is showing the percent increase in genetic gain in the mean that you obtain from using D-cross relative to selection on the mean only. And so on the y-axis, it's the increase in gain I get, expressed as a percentage. On the x-axis, I've got the number of environments, and each line is representing what is our target for stability. And so if I'm trying to create products that are in the top 1% for stability relative to the parental population, using this D-cross strategy will give you close to 10% more gain in the average performance across environments relative to if I just selected on the mean alone. And so you can see as that threshold and that requirement for stability decreases, the relative increase that you get decreases over time. But even with quite small thresholds and requirements for stability, you still get this gain in overall performance. So what does it look like relative to if I just did a penalized index? So that's the other approach, and so you can see here that the gain that you get is nowhere near as favourable if you were to use this crossing strategy. So it's a lot better to consider the outcome in the progeny rather than penalising stability within the parents. You get considerably less gain in the products. So just to wrap up with some conclusions, so genotype bioenvironment interactions can be useful in breeding programs, and they are useful, and we can use them to create stable products for release. These are applicable to all types of breeding programs. I was actually using an inbred breeding program, but this is probably easier to implement in a hybrid breeding program. So the real message is unstable genotypes are valuable and shouldn't be discarded in selection, and we can use them in breeding programs. And there should be a lot of thought given to actually how we utilise this variation in breeding programs. And we're working to validate these results in real data. And so I'll just finish by thanking Bay Crop Science, the BBSRC, and my co-authors for funding this research and helping make it happen. Thank you.